So okay. Oh. So replicates and then condenses. The nucleus membrane and the nuclei begin. The nucleus begins to like fade away or dissipate or break down. Spindle. I'm sorry. Centrioles move to the polar ends of the cell, and spindle fibers begin to form. The spindle forms and binds to the chromosome pairs. Okay. Then in anaphase, or rather, the chromosomes line up in the center, forming a metaphase plate, and then they bind, and then in anaphase they divide, or the spindle fibers begin to contract. But as a result, they don't actually split the chromatids into two separate chromosomes, but they actually just move. They break apart the pairs into separates. So now, what you're left with is one chromosome, which is a pair of chromatids. From each homologous pair moves to the end of the cell, and as a result, the chromosome number in each cell is half of that original. Oh. Now, I think the area where you have variation is where it lines up whether the mother or father is on the left or the right is completely random. For sure, that's where one form of variation is. When the nuclear membrane reforms and the cells begin to divide by cytokinesis, remember that there is going to be the contractile fibers. It's okay, it's okay, it's okay. It's okay, Amo. It's okay. So, in some cells, this continues to full cytokinesis, and there may be a period or brief prolonged interphase between this first phase of of meiosis and this second phase of meiosis. But ensure that there's no be aware that there is no further replication or S phase in that in the time between these two. Whoa. There's no S phase in the time between these two. Okay. Then when we go to the next metaphase, we see that each of these. Also, I want to describe what cytokinesis is real quick. So this you have the contractile ring fibers which pull or contract, causing a cleavage to form. Sorry, a furrow to form. Then as the cleavage furrow becomes tighter and it essentially pinches between the two cells, which then forms two separate cells. Okay. Now metaphase 2 is where new spindles are formed. It's essentially they are already lined up, so we don't have to do prophase. Okay, we just go to metaphase. We don't do P. We just do. If we just write down this, we just do MAT because prophase is meant for condensing. That's already done. Okay. Then metaphase is you line them up. So you can say that the chromosomes line up on the plates, and the spindles are already on the polar ends. So not spindles, the centrioles. And they form spindle fibers which bind to the centrosome or centromere. they don't say. Oh, I can do this. Okay. So, yeah, they bind to the. I'm pretty sure this is the centromere. So when they bind to the centromere, then begin to pull. Because like the binding is happening at metaphase, and then they pull in anaphase, which splits the chromatids apart into chromosomes, right? And then we have, as a result, we have cytokinesis occurring, which is going to is going to separate these two cells into four haploid cells. The reason we call them haploid is because it's half of the original cell, which was a diploid, right? Why was it diploid? Because I'm not sure. Because I guess it had like an even pair. No. Hmm. Not sure. Anyway, so then these unravel. So this is telophase two, where I would say the telophase two should actually be like in between these two spots. So you have like this kind of forming, right? So telophase two is where the nuclear envelope reforms, the nucleus reforms, chromosomes unravel, and go back to interphase state. Cytokinesis occurs, giving four daughter cells, each with half of the chromosome number of the original diploid cell, which is why we call it haploid. The original cell was diploid, and if we have half the chromosomes of the diploid, we can call it a haploid. Now, what do they mean by crossing over, right? So that is, I hope, yeah, they talk about it here. So meiosis and genetic variation. Oh, wait, before we continue, um, I guess I could do this. Okay. So meiosis reduces the chromosome number in gametes from diploid to haploid because this is half of the original diploid cell. It is the main way by which genetic variation occurs in species. This is pretty important. I mean, it sounds pretty important. All like the like evolving or like the whole evolution process occurs because of genetic variation, and genetic variation is only possible, or it's mainly possible because of this specific you know process of which we produce our sex cells, right? So this variation is introduced by mainly two processes. One is independent or random assortment, and crossing over and recombination. So crossing over is the exchange of genetic material between the non-sister chromatids or of the homologous chromosomes. So crossing over results in recombinant chromosomes. Okay, so this I've heard this term and I'm not sure what it is, but recombinant chromosomes. Cut and stitch bits of maternal and paternal chromatids together. The points where chromatids break are called the chiasmata, and the swapping sections of chromosomes gives the recombinant chromosomes. So just to show you here, if we have the maternal and paternal chromosome, and these are homologous pairs, right? And so this is where, like, you would imagine in metaphase, they're lining up like this. So this is from the dad, this is from the mom, right? So what ends up happening is that at points where they make contact, this is where the chiasmata. So let's say they move closer. Um, how do I do this? So this is from the dad and this is from the mom. So let's say at one point it moves closer and it touches like over here and over here. So the chiasmata would be here and down here, right? So at these points where they make contact, the chromatids will break and rejoin, and the non-sister chromatids will exchange the corresponding sections of DNA known as crossing over. So over here, if they touch right here, so then essentially they touch here and they touch here. So then they will swap uh, the genes over there. So this section of DNA came from the dad, this section of DNA came from the mom, right? So they swap there. Um, they cut and stitch bits of maternal paternal chromatids together, okay, or the genetic information. So that when they separate, right, and then these break apart, you can see that over here you have one pure, one kind of different, one kind of different. All of these are different, right? None of these are the same. And none of these are the same to the original parent either, right? So, yeah. Now, crossing over can result in a great deal of genetic variation. Crossing over produces chromosomes that obtain new combinations of alleles for both parents. Independent assortment is the random distribution of maternal and paternal chromosomes into gametes during cell division. Chromosomes are. Is this what I meant? Yeah. Okay, nice. Chromosomes are from the parents are distributed randomly into the gametes. There are more than 8 million potential genetic combinations within the sperm and the egg. So, gamete producing cells, other chromosome arrangements are possible with the 3 pairs of chromosomes. Wait, what? Yeah, okay. So when this divides, you'll have pure, and then kind of weird, right? So this is because how they lined up. And then during, what's this called? At anaphase 1, you'll be separating these apart, right? So this is where what these two separate cells would be containing, the daughter cells. Now, when this then undergoes meiosis 1, you're gonna see that like you're gonna have loads, right? So yeah. How meiosis produces genetic variation in gametes? So independent random assortment and crossing over, these are the two that we learned. And independent assortment gives rise to new combinations of paternal and maternal. Crossing over involves swapping of sections of chromosomes which produce recombinant chromosomes. Okay, so remember the difference between the two. Independent assortment gives rise to new combinations of paternal and maternal chromosomes, whereas crossing over involves swapping sections of chromosomes, which produce these kind of special altered chromosomes, which we call recombinant chromosomes. Okay? Importance of meiosis and gamete formation. If meiosis halves the number of chromosomes in gametes, so the diploid condition can be restored after fertilization. Right? So we need the importance of meiosis. Like we can't use mitosis here because we need the meiosis to have to be a haploid, so that at fertilization, the haploid and the haploid of the female, the haploid of the male and the haploid of the female, can combine to form a diploid whole cell. Okay, so this is why meiosis is super important here. Okay, meiosis also leads to the genetic variation in gametes by crossing over. So it's not only that you need to have the proper amount of chromosomes, but also you need to have some genetic variation for evolution to occur. Now, let's look at the specialization of gametes real quick. All I really need to know is the tail. Lots of mitochondria stored here, the acrosome and the nucleus. That's really it. What did I say here? I said mitochondria, right? Yeah. So that's pretty much it. Like I've learned this like 1,000 times. I feel like. Um, Several mitotic divisions occur in the production of the sperm cell. To increase the number of primary spermatocytes to produce large number of sperm cells. So, um, so the production of sperm cells is almost the same way you would imagine the reproduction of, because these are unicellular, right? So if this is unicellular, you can imagine that when this divides in the same way, it's going to be through mitosis, right? So mitotic divisions occur when we try to produce more of these cells to increase the number of cells to produce a large number of cells. Well, sites, which are enzymes, right? Or spermatocytes sound like an enzyme. So we can see by mitosis we form this site, and then by meiosis we form the cell. Sorry, this is a gamete, isn't it? My fault. So this isn't through mitosis, it's through meiosis. But we increase these by mitosis, which then by meiosis gives us the thing. So the original cell is highly, like, it's made in lots of amounts through mitosis, and then each of these undergo meiosis to produce the actual gametes. So streamlined shape reduces resistance. Acrosome contains acrosin, which is a digestive enzyme to break down the zona pellucida. Haploid nucleus restores the diploid chromosomes, right? It's already haploid because of the meiosis that occurred. We could also say, actually no, that's fine. Large number of mitochondria to provide energy or to release energy for movement, providing, uh, for movement from the tail. Flagellum allows for motility or enables motility to receptors in the cell surface membrane to bind to egg cell surface membrane. Okay, I did not think of that one. So you have receptors over here because you're gonna have binding sites on the, like you would imagine you would have binding sites on the egg cell, so egg surface membrane. So you would also imagine that you have receptors over here as well on the cell surface membrane of the sperm cell. Now, when we look at the human egg cell, this is also something I kind of forgot, but anyway, I do remember that there was like a very big thing on the edge that they're not showing here, which is weird. Yeah, this thing, that's like these spiky bits. Um, so anyway, um, you're gonna have the cytoplasm, you're gonna have the nucleus. Remember, this is gonna be a haploid nucleus, obviously. You're gonna be having lipid droplets, no starch droplets. You're gonna have lysosomes. Why? Um, Also, the process by which this is made. Remember, we call this an, how do I pronounce that? I oocyte. Oocyte. Oh, it's literally called oocyte. Oocyte. Okay. So by mitosis we form the oocyte, and then by meiosis we form the ovum. Okay. Is the ovum? So by mitosis again, you form the spermatocyte. So you have the oocyte and the spermatocyte. This is the spermatocyte is made from the spermatogonium, whereas the oocyte is made from the oogonium, right? So should be pretty easy to remember. The only difference is this is an ovum and that is a sperm cell. Okay. Um, Now, what else? So you have lipid droplets, you have lysosomes. Then on the outer edges, you have uh, Yeah, you have the cell surface membrane over here. So you can imagine some kind of like bumps and stuff for binding. You have a jelly-like coating, which is called the zona pellucida. Now, jelly-like coating, maybe you can think of this as the capsule in prokaryotes, but I don't know. And then you have follicle cells from the ovary. Okay? So from the ovary, you have follicle cells. Okay. Now, I don't think it's necessary to know that it's from the ovary, but it's extra information. Now, haploid sets of chromosomes in the nucleus, that's obviously has to be present so that you have a diploid cell on fertilization. Lipid droplets, which supplies energy for division, because division is going to be occurring here. So note that this is the site of cell division. It's not over there, it's here. So the sperm cell needs energy for traveling, whereas the ovum needs energy for the actual cell division to occur, right? So that's what the lipid droplets are for. Large cell size increases the surface area, so you have a greater chance of fertilization. Cortical granules. Now, cortical granules are okay. It's in the cytoplasm. It's not labeled here, but you can imagine there are cortical granules here and there, which help to prevent polyspermia. Okay, let's see. What are cortical granules? কোথায় যাচ্ছ? Oh, it doesn't look really pretty. This diagram makes no sense. What's going on here? What? The insulin diagram. The what? Yeah. I get the thing, but I just don't understand. So, uncontrolled amount of glucose comes in. This is an endpoint, right? The blood glucose level needs to be reasonably constant. These are like valves essentially, which then can control how we can remove or we can add in glucose so that we can keep the blood glucose level constant. So glucose comes up here. So what are they doing? Glucose levels get very high after a meal, but all excess glucose is excreted. And that means okay. Okay. I know your house. I know you don't, but I know you can. Mama, you need to do exercise, otherwise you'll get type 2 diabetes. Please, no. I'm actually serious. Stop! I don't want to get diabetes! Type 2 diabetes is common as people get older and it's often linked to obesity, lack of exercise, or growth. Please. Literally, it's just not my fault. That eraser wasn't very big. What was that? It's just. ক্যাপ লাগানো একটা পেন দাও তো this one and give me a I'm gonna give you my favorite pen, okay? But you gotta be careful. No, no, I have to I'm gonna have to pen 9. Why is the stylus not much? Someone's never been got to go to it whenever you ever. You never did that with your brothers? Ever? Or your brothers never did that to you? Oh. Said is that in the Quran or something? I don't know. No! I don't want to be an adult. No!
I can't see it. It's everywhere. I really not see it. I can't see my glasses. Father, did I bring the father? শুনে যা। আমার মোবাইলটা ঘুরাচ্ছে। মোবাইলটা ঘুরতে অনেক extra লাগে বাবা। Father, did I bring the files? Allah. It's not there. It's not there. Feel ashamed. একটু যে আমাকে দুই না করতে ফাইল রেখেছি টাকা বলবো টাকা বলবো ওষুধ দেখুন মনে হচ্ছে
Can I please go to the living room?
Huh?
Can I please go to the living room?
What did I do?
You guys are just talking a lot. And I'd like to sit somewhere comfortable as well. I mean, what's so yeah. Film if you want, you can sit at the table. Help me understand. Here's how the particle granules to prevent body spawning in the cell after fertilization. Fertilization.
আমার মনে হচ্ছে যে পরে পিছনে হ্যাঁ হ্যাঁ দেখে যদি না থাকে তাহলে আমি এখানে আবার ভালো করে খুললাম একটা দিয়ে
Do not cover a breathing hole. Oh, look at the graphic design here is kind of crazy.
কেন আমি শুনে এটা আছে বলে একবার ঢোকাতে ঢোকাতে পারলাম না ঢোকাই দিলাম পেশেন্ট মনে হলো দিয়েছি না
One sec. Okay, now that's done, because I have to do that every day once. Um, let's just work. Hashtag neat, hashtag hikikomori, hashtag anime, hashtag ASMR. Anime ASMR. Oh, that's genius. $1 billion idea, bro. How'd you be able to capitalize? So true. Alright, cholo. Boop, 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 boop. Ba, ba, da, ba, 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 da, da, da. Ba, 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 da. Whoa, this looks nice. Do, 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 do. Okay. Special mechanism designed. Oh, it's the thing that like hardens after the first penetration, right? Catastrophic fertilization of an egg by more than one. If multiple were to, then you would possess an abnormal amount of chromosome sets while a ploidy. rendering it unviable. Cortical granules ensure that does not happen by orchestrating the slow block to polyphony, which falls in a precise. The moment a single sperm successfully binds to the egg's plasma membrane and fuses with it, it triggers a signaling cascade, causes the whole thing. Uh, from the eggs endoplasmic reticulum. Oh, it does have that. Well, we didn't draw that. I mean, I guess if there's a nucleus, then you'd imagine there's an endoplasmic. This is also we saw over here. Okay. Bush mRNA to present to allow early translation of transcription factors. So in its DNA, there's wait, what? How do you already have mRNA present? I guess you okay. Okay, that makes sense, I guess. Yeah. Structure differences between sperm and egg cell. So flagellum is in sperm. Sperm is streamlined for movement, whereas ovum is round and larger for more surface area contact. Fewer mitochondria in sperm, which is more in ovum. Oh, really? Okay. Um, acrosome, zoonoplasuda, jelly-layer, cortical granules. Sperm has less cytoplasm, ovum has more. Okay. Similarities is both they contain nuclei, both are haploid. Both contain a nuclei, nuclear pore, nucleolus, double membrane. Both are produced by meiosis. Released from the cells surrounding the ovum, triggering the acrosome reaction. Lysosomes in the ovum thicken the jelly-like layer, preventing entry of the sperm. Nuclei of the ovum and sperm fuse. Jelly there, follicle cells, lysosomes. What releases the things that attract sperm? The follicle cells of corona. Bro, my laptop is struck like 1 second. Oh my days. What is up? What's going on? Do do do. You guys give me energy. I'm 27 years old, no degrees, no friends, boring job, always thinking negatively, but now I'm gonna start again, quit social media, find my passion. Unironically sat in my chair staring at the ceiling for 20 minutes listening to this. Rookie numbers, rookie numbers. Oi, oi, oi. Paranoia, man. Paranoia. It might sound odd, but if you're in a big city, hit up your local IATSE stagehand union. See if they have a stand. See if they need a standby for a pusher or carpenter on the next Broadway show. If you have that in your town, it'll help you get out of your usual work culture for sure. Might not even get you more work plugging cables in or turning a wrench on some truss. Bro, what the hell? Okay. Hmm. When you take off your headphones, with the purr 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 and doesn't stop. That's funny. No, your need opportunities it's hard to like to listen. uh, man, that is crazy. Life is crazy. Okay, so plant cells we don't need to do anything. Flowering plant cells. So I mean flowering plants. So we can just kind of we can just keep on going from here and just stop here. And then I think we go into that stuff. Yeah. Let's see. Yeah, so this is kind of interesting. So we have uh, the chemical released triggers the active reaction. It's not necessarily actually action swells fuses with the sperm cell membrane and releases actinosin. So sperm cell then releases actinosin by excitosis. The enzyme digests to the follicle cells and the jelly-like layer, and then eventually fuses with the ovum membrane. The sperm nucleus enters the ovum cytoplasm. Once the sperm nucleus enters the ovum, meiosis II is complete. So, wait, what? It's doing. Uh, oh, okay, no. Okay, ensure that meiosis II is done by that time. Okay. Coral granules move and fuse with ovum membrane. And wait, I'm so. Meiosis II has to be done because one sperm cell is one haploid of the four haploids made from the informed development cells. Two haploid nuclei form fuse and forms a segment. Yeah. Okay, sweet. So now that's done. Genetics is going to be the next topic. But before we actually touch that, let's just quickly go over this. So here they're talking about nervous system. We have a note on nervous system. Where is it? It's unit 5. Is it 8A? Alright, let's go. Come on! Okay, so we've already done this all before. I've actually recorded all of this already, so this should not be too difficult. But I do kind of want to like get through this, and then kind of see like based on what she wrote down, like what do I kind of need to know. I mean, if she got in, then everything that's in there, I should kind of like get an idea. So um, that's one thing. So they kind of touch over what? So they touch over first the structures, then the speeds, then the resting potential, how the resting potential is maintained, how the actual potential is produced, and what happens afterwards. Effect of a different stimulus on actin potential. Um, right, I remember this, right? So I forgot what it was called, but it's like the potential difference that which doesn't reach this threshold, it just goes back down, right? There was a name for that, I forgot. Refractory period, propagation along the thing. I'm pretty sure this is how it travels, right? Um, Saltatory conduction, I do not remember this. Like, this is how it jumps, right? Yeah. Then synapses, this is pretty easy. Neurotransmitters, I'm pretty sure she mentions some neurotransmitters over there, so that kind of gives me an idea of like which neurotransmitters I should know. Acetylcholine, dopamine, is that glycine or something? And noradrenaline. Um, let's see. Types of neurotransmitter receptors. Yeah, I'm pretty sure that's also there in our thing. And but that's cool. Neurotransmitters are the can't read that. buddy. Terminal within synaptic vesicles. Okay, yeah. That's the presynaptic, right? So postsynaptic cell could be anything, another neuron, muscle cell, gland. Oh, right. Okay. Wait, really? A muscle cell? Um, I suppose, right? Because I mean, if the neuron was an effector cell, then yeah, right? Gland, etc. Right? Muscle cell and gland. Those are usually what the effectors are we kind of connected to. The gap between the presynaptic membrane and the postsynaptic membrane is called the synaptic cleft. Yeah, that's correct. The action potential that reaches the axon terminal calcium ion channels open, allowing calcium ions from outside to enter the membrane, which allows the synaptic vesicle to fuse with the. Was she reading from my notes? Because I'm pretty sure I sent the. Did I send those to her? I did not. I don't remember. I don't remember. No idea. Honestly, it doesn't reach up there, does it? Okay. I mean, sure. I have to like very like make sure that these are like stamped down. Pretty sure. Yeah. Mm, the drug can now diffuse across the synaptic cleft receptors and the synaptic. Depends on the action potential of the frequency. Okay. I don't really remember how the action potential works, so I'll need to. And then it goes and talks about circulation a little bit, and then brings that back to this thing. I don't quite remember. So let's ignore that for now. Okay, so most of this seems pretty basic. So let's just get through this as fast as possible. So, so we have neurons. Neurons are going to be our nerve cells. Okay, nerve cells are known as neurons. They're going to be structural and functional units of the nervous system. So how we have muscle fibers, those are the structural and functional units of our muscular system. I don't know if that's what it's called, but um, yeah, the nervous system has two parts. We have the central nervous. Oh God, man, maybe I should revise 7A, 7A BC, because I feel kind of bad now. Not 7A, but 7B and 7C. Just look at all of those, right? And like the type of different muscle, not muscle fiber, but a blood vessel. That's unit 1. So like that's far back enough that I don't really need to care that much. Oh my God, look at all this. This is on that web browser, right? Yeah. Okay, this is honestly quite short, and it's also really good because it talks a little bit about this, which I know she probably has written a lot more about, and I would like to see if it goes more in depth. Then this cycle, protein metabolism, as well as the excretory system, this is huge. Mm, yeah, this too. And then yeah, ADH and FSH, right? FSH is here, is it? I don't know. Actually, is it not? I don't know. Then I remember for where is it? There was I guess it's an 8B and 8C. Yeah, yeah, yeah. Because they talk about homeostasis in the next few ones, right? So where do they talk about the lungs, by the way? Like the cardiacs and the lungs and all that. Is in the next few. Oh, okay, this is all GM stuff. Okay. Oh wow, wait, what? Recombinant DNA? What was the other thing? Recombinant chromosomes? Wait, that's funny. Huh. Right, because you are switching, you are like inputting, or you're like swapping some parts of the DNA. Huh, that's cool. Okay, that's actually so cool. Huh. Alright. Um, yeah, this is pretty big. Like knowing this, and then they're gonna bring it into. There's this. Is it not here? It, it was in the last unit. Wait, what? I'm so confused. Where's the? Is it here? I wish I had my textbook because I just don't know. Um, I do have it. I mean, yeah. Let's see. Oh my god. Okay. It uh, it's 7B. Yeah. Principles of homeostasis. Oh, it is here, isn't it? Yeah. Okay. I don't know how I missed this, but that was weird. Okay, so this is probably like gonna be the hardest document, like by far, like by far. Um, this leading into 7C is gonna be pretty tough. Uh, at least 7C isn't too bad. Like there's a lot of info, but it's not too bad. Because I'm pretty sure this brings into the eyes as well, or is that eight C 8B? Because that also talks a bit about like it's not really homeostasis, but it's like reflex arc. So it's like the subconscious stuff. Yeah, PFR. Okay. Wait, no, PFR is in plants. Um, where's the eye one? It's cranial reflex. Where's the cone cells and stuff? Forgot what it was exactly. Ah, there you go. We got a lot of reading. A lot of reading today. What should the order be actually? Oh, yeah. Because actually speaking, if 7B talks first about like initially talks about homeostasis, then we might as well do 7B first. Because these two feel like they're important for this. You know what I mean? Just the only reason I wanted to bring this up first was because because there's a picture of it first. But that's maybe that's not a good idea. Okay, fine. Then I'll take out my phone and I'll just charge it just in case I need to like review with what she wrote. Uh, That's so much work. I mean, honestly, I feel really good though. Like, I'm glad I'm getting that revision, bro. It's been so long. It's been too long. Okay. Is there anything else I want to do before I kind of start? We'll start off with 7B. What does 7A talk about? It's that, which is not really related. So we can start with 7B, I think. Right? Right? 7B. I don't wanna. Uh, I don't want to. God damn it. Oh yeah. Also, she was talking about like like I guess they might ask like how to draw things, right? So that's another thing. Um, because I don't really remember how these look. I mean, striated or whatever. That might be the word. Yeah, not striated. What is striated? Is it repeating or something? Or it has stripes. Okay, striated coming from stripes. Okay. And then also like this sarcomere and like being able to understand the what's it called the sliding filament mechanism. I hated this one. So it was just like hard to explain. This was all just very just confusing. I kind of just guessed through it. This too, like no idea what was going on here. This is a cool drawing. I I did this in school, or I don't remember when I did this. I did this in school. Yeah. Do I have a info on this? Like properties? Oh my God! Look at that. Created 17th of February at 10:23 a.m., which is probably 8 o'clock. If it's talking about my time. So yeah, this is in school in the morning. Yeah, I remember this. But anyways, yeah. Okay, so then B part two. Then they talk about the internal environment. So these are two pretty different things. Actually, I would rather focus on this second half because it deals with homeostasis, which I can connect to this, which I can connect to this. Um, but let's keep it simple. Let's keep it simple. Um, it might be the white. Why is it charging so slow? Oh, there you go. Hopefully I can read on the phone, so it doesn't cause too much problem. Alright, let's just start. If I want to switch back, I can just switch back anytime. It's okay. So, neurons are the building blocks or the units of the nervous system. The nervous system has two parts: the peripheral and the central nervous system. The We already know that when we have like a sensory organ or a receptor organ, then we're going to be having sensory neurons, which carries the impulse, which is generated by the receptor, to the CNS. And then the relay neuron connects the uh, neuron to the right. Synapse connects these two, but it's the relay neurons in here which then which then allows the impulse to be moving to the motor neurons, which go towards the effector, from the CNS to the effector. Now, neurons, which are nerve cells, consist of nerves, which consist of nerve fibers. Okay, so keep this aware where when we say neurons are nerve cells, right? So nerve cells are known as neurons, which are the building blocks of the nervous system, or the units of the nervous system. They consist of nerves, right? Which are consisting of nerve fibers. So we have one big nerve cell like this, say, right? And it consists of a bunch of nerve fibers, which each all consist of, no, nerve cells consist of nerves. Nerve cells consist of nerves. So let's say this is one nerve cell. This is consisting of nerves, each of which consists of many nerve fibers. Okay? It's pretty much it. I don't know how much I'm supposed to know from the structure. I'll just look at what she wrote and just kind of guess upon that. So now it consists of nerve fibers, which are the axon and the dendrons. Okay. So wait, what? Wait, what? Wait, what do you mean? Okay, let's just get the idea from. So first of all, can we make this? Okay. Okay. So let's get the eraser in here. So help me understand. Okay. Let's see. Is it a jet? What? Alright, I don't know why I've been spending more time on that server. It's so weird. Like just randomly. I've been just like appearing on that server more. Really weird. And unexpected. Okay. So over here, they're just talking about the structure, Myelin sheath. Uh-huh. I don't know what that says. I haven't seen anything she wrote here that was extra yet, so unless, let's see. Um, yeah, everything from the thing. Okay, yeah, everything from the nervous system seems to be pretty important. It's so cool to see how different people study. Like genuinely, it's so interesting. Because it's like I do not write notes at all. It's so funny. Um, I wonder if that's a bad thing. I don't know. I've always been taught to not write notes, you know? Which is like, I mean, I understand writing notes based off what you remember. That's good. That is, that I should do. That I don't do. That I should do and I don't do. Yeah. Okay. So individual specialized nerve cell. Okay. Consists of a cell body which is a soma. Oh yeah, I forgot about that. Containing the nucleus and the cytoplasmic extensions called dendrons. And an axon. So the main parts are the cell body, which contains the nucleus, as well as extensions called dendrons, which receive the signals, and axons, which transmit the signals away. So the cell body, the receiver, and then the transmitter, or like the shooty guy. Shooty game. Nerve fibers, which are the subcellular levels, with a long. So neuron is the same thing as a nerve cell. So it is a specialized nerve cell. Interesting. Nerve fiber is a long, slender cytoplasmic extension of a single neuron. The term most frequently refers to the axon. Okay. If a neuron is an entire electronic appliance, then the nerve fiber is just the long copper wire protruding out of it to transmit electricity. The nerve is a complex macroscopic structure found exclusively in the ENS, solid cord-like bundle containing thousands of individual nerve fibers, which are axons bound together alongside blood vessels. Inside a nerve, uh, nerve is like a massive underground telephone cable containing thousands of individual insulated copper wires bound together. I'm still a bit lost on the terms here. Okay. Sensory neurons carry the neurons which consist of nerves, which consist of nerve fibers. And nerve fibers are the axon and dendrons, and neurons consist of nerves. You said nerves were the organs which have like loads of oops. Nerves were the organs which contain loads of stuff, right? Which makes no sense. How does the nerve cell consist of the nerve organ? I don't. Does a nerve consist of neurons? I think they're getting azan. Need to go pray. Nerve consists of neurons. Oh. Okay. Yes. Okay. 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 Yeah. Okay. You get the idea. Let's see. Let's look at the. I think it would be nice if I could learn to actually draw these just in case. And also, I mean, why not? I like drawing. Let's see. Where's my thing? So, do I have a pen? So, we have three types of neurons. We have sensory relay and motor neurons. Remember, nerves consist of neurons, and neurons consist of, since neurons are the individual cell, neurons consist of the axon dendron, which those are the nerve fibers. Okay. Now, so these are the nerve fibers. This whole thing is a neuron, and then I would guess as this like elongates to like loads like a whole thing, that might be a nerve. I'm not sure, right? Because when we see that whole line on a skin, we call that a nerve. We don't call that a neuron, right? So and that is made up of many that is made up of many neurons, which are like the units of the nervous system. This is just how I'm gonna understand it for now. The three ones, okay, let's just quickly understand. So let's start off with the sensory one first. So we have, remember that the axons and the dendrons, right? So the axon, we have the cell body, the axon, and the dendron. And so the dendron, okay, what did they say? Let's just write this down first so I don't forget. So we have cell body, axon, dendron. Okay, axon is meant for, I swear it just said it. It's over here. Yes. Dendron receives, axon it transmits. So dendron is the receiver and axon is the sender. Okay? Cool. So now over here when we look at this, we see that the axon is here. So we know that for sure it's gonna go, yeah, it's right here. The direction is gonna go this way, right? So we see the cell. Okay, let's look at, since we know the main parts is the cell body, the axon, and the dendron. Let's look at where they are. So cell body is kind of in the middle. Okay, cool. The axon is on the left and the dendron is on the right. Okay, cool. That's it. That's the main thing. Now let's look at a little bit of some of the features. The axon has synaptic bulbs which pass on the axon has synaptic bulbs which pass on the impulse. Okay, cool. So it has synaptic bulbs. Synaptic bulbs give you the idea that there's going to be a synapse next to the next neuron. Maybe this is where the thing goes and the neurotransmitters are released, something like this. Okay, cool. Then We have node of Ranviers. Okay, so this is gonna be some basic stuff. So myelin sheath, node of Ranvier is the gap in the myelin sheath. Then okay, we also have Schwann cells, the nucleus of Schwann cells, and we also have the dendron. Okay, so what is the difference between a dendrite and a dendron, right? So when we read here, let me see. You said what is the difference between dendron and dendrite? It seems. I mean, to be honest, I mean, I guess dendrons are receiving it, but then it's also sending it forward. So isn't these dendrons technically like um, acting as both transmitter and receiver? Distinguish the distinction comes down to anatomical size, direction of branching, and specific type. Dendron is the main trunk. From the cell body, it acts as the main conduit traveling toward the cell body. Alright, because we have dendron and then we have, what's the other one called? I actually forgot this entirely. Wow. What's the thing called, like, where it leads up to the cell body and then everything after it? Oh, I see. Okay, so it's not just the front and the end. Oh, and it says right here that they're nerve fibers. Okay, this is cool. Okay, that's interesting. So it's the whole thing that leads up to it and the whole thing that comes out of it, right? So essentially the dendrites kind of branch out into dendrites, but after they kind of join together, then there's a literal straight dendron which leads to the cell body, and then after the cell body, it's a straight axon which then kind of turns into the synaptic bulb. So how the dendrites are not really the dendrons, the synaptic bulbs are not really the axons in the same way, right? So that could be our little way of understanding that. So yeah, um, let's look at the last thing that we haven't looked at yet, which is this, right? So this is going to be the receptor, which is going to be a receptor for pressure, a receptor in the skin. You know, we have baroreceptors, we have pH receptors, we have pressure. I'm pretty sure it's the same baro. We have carbon dioxide levels. We have um, sugar levels, right? We have I don't know what else we have. Also, dude, we have to learn immunology, bro. Immune system. That's gonna be crazy. Immune system is gonna be crazy. It's gonna be crazy. We have so much to do today, bro. I'm kind of excited. All right. Um, how long is this stream gonna be? Like for real? I've already wasted a lot of time. I understand, but like, I haven't even gone that far yet, and I'm already like, holy shit! Like, this is gonna be a long stream. All right. Let's see. Let's get a blue pencil going, or actually a red pen is actually probably better. So we know for sure that we have a receptor over here. And then we have the dendrites, which join together to form this one dendron, leads to the cell body, which is I drew really badly. It's like up here, and it goes up. Then it keeps on going to the axon, and then it splits into synaptic bulbs. Okay, so now we can just draw each thing like this. Okay. This has a nucleus, obviously. Okay, we don't really know specifically what a Schwann cell is, because I thought those were sheets. I thought that was like a slimy capsule kind of thing. But it says that it has a nucleus. So is it a cell? Like I'm kind of interested now. What are you? Okay. Besides that, okay, tell you what, I think it's better that I open all of this on my phone, so I don't have to um, what's it called? Like leave the screen because I just don't want to. I'm scared. I don't want to do that. So Okay. Okay. So okay. Are we done with the drawing? So it's really ugly. I just want to let you guys know. um, Dendrite's receiving the impulse, keeps on going that way. We have a cell body in the center, and then I think the most important part is knowing where the cell body is, where the axon is, and where the not really where the axon and where the dendrons are. I mean, it kind of, but also more importantly, like because I mean, if you know where the cell body is, then you know where the axon and the dendrons are. That's the thing. Right? Okay. Now let's look at the motor neuron. I'm not gonna really talk much about the Schwann cell or the things because it's not really that important right now, right? So next one is gonna be this, and we see that the cell body is all the way on the right, okay? And therefore, we know that if the cell body is over there and the axon goes away, transmitting, so then it's just gonna keep on going to the left, and then there are no dendrons. It's just directly dendrites, which are receiving the impulse, and then immediately gets sent to the cell body. There's nothing in between the dendrites and the cell body. Whereas over here, since there was, the dendrons were present. So axon will bring them all the way straight to here. Okay, wait 1 second. These are basic myelin sheath, Schwann cell nucleus, node of Ranvier's. This is going to be common. And then on the end, notice how the axon splits into two, and um, we have these synaptic bulbs, and they lead straight to the muscle. So this is what Ajay was saying. Right? She said that the synaptic cleft will be between the presynaptic neuron and um, and anything else. It doesn't have to necessarily be another neuron. It can also be a muscle directly, or it can be a thing directly, a gland directly. And I actually didn't realize that, but it's right here. Also, it's not just there; it's also here, right? Because there's also going to be, well, this is not really the same thing. How does it receive it here? Is there a synaptic cleft here? Okay, actually, I'm going to ask that. So if there is a synaptic cleft between the motor neuron and a okay, between the axon, the motor neuron, um, synaptic synaptic bulb, and the muscle for for the impulse to get transmitted. How does the sensory neuron receive the impulse from the uh, receptor organ if there is no synaptic bulb slash cleft on bulbs on muscle cleft between them? The transmission of a stimulus from the receptor organ to a sensory organ operates on a completely different structural mechanism than the transmission at a motor neuromuscular junction. Motor neuromuscular junction. Okay. While a motor neuron uses specialized synaptic bulbs to release the neurotransmitters onto a muscle, sensory neurons use a specialized sensory receptor embedded directly at the terminal ends to convert environmental energy into an electrical signal. This is how it works without a traditional synaptic cleft. So transduction instead of synaptic transmission. At a motor synapse, chemical signals are converted back into electrical ones. At sensory receptors, this process is called transduction, where the conversion of a physical or chemical stimulus, such as pressure, heat, light, or chemicals, directly into electrical potential. Sensory neurons do not require presynaptic bulbs because their dendritic endings are modified to act as sensors themselves, which happens via two main structural designs. Modified free nerve endings. Dendritic endings of the sensory neuron are naked or encapsulated directly within a non-neuronal tissue with no synapse involved. When mechanical pressure is applied, these layers deform, physically stretching the plasma membrane of the neuron's tip. This mechanical stretch forces the stretch-gated sodium channels, allowing Na+ to rush into the thing, which generates a potential. The second thing is epithelial receptor cells, which is specialized sensory organs like the retina or taste buds, which the receptor is a distinct specialized epithelial cell, not a neuron. So the sensory organ has the receptor is a specialized epithelial cell, not a neuron. So the stimulus, which activates the receptor cell, causes it to depolarize. Once depolarized, the cell releases a chemical messenger across a localized gap to activate the adjacent dendritic endings of the sensory neuron. So it's kind of similar. The main difference is in motor neuron endings, an active center of signals. It requires synaptic vesicles and a terminal bulb to release chemicals across the cleft, whereas in sensory neurons, you have a passive receiver or a transducer